Agnes von Matuschka, Potsdam Science Park.
Thank you. Good afternoon. My name is Agnes von Matuschka. I'm manager of the Potsdam Science Park. It's a biotech and life science park just next to Tesla Gigafactory. We have Max Planck and Fraunhofer institutes and new lab space that we rent to interesting companies. In 2021, biotx came to us, a very international company, with Marco Schmidt, and they settled here, and it's quite impressive because they came during COVID pandemic situations, and they implied their new technology, their artificial intelligence, to predict the drug efficacy in clinical trials. And they are successful, and I'm very happy that they're here and being selected to present their technology. Thank you.
Breaking the wall to synthetic clinical trials. Marco Schmidt, biotx.ai.
Good afternoon. My name is Marco. I'm a biochemist, and I'm one of the co-founders and chief scientific officer of biotx.ai. biotx.ai has 3 drugs in development licensed, based on validated AI-enabled causal models that predict a high likelihood of clinical success. Currently, we have one repurposed drug for severe COVID-19 in a phase 2 clinical trial, and we have another repurposed with a new formulation, with a patented new formulation, drug for Behçet disease, a rare autoimmune disease, close to entering a clinical trial we are now doing fundraising for. So the question, why we think we have a high likelihood of clinical success. The point already is, we know that drugs fail in clinical trials due of a lack of efficacy. So in other words, we picked the wrong drug target. From retrospective analysis, we know that when the drug, which is going for a drug target, the drug target is genetically associated to the disease, the drug is 2 times more successful in a clinical trial. And this is association. We at biotx.ai, we go for causation, we go for causal models. So how does it go? So we are solving 2 problems. The first one is that genomic data in particular is so-called high-dimensional data. So you have much more genetic variants than individuals in your data set. So we have built a feature selection tool which enabled us just only to select the important genetic variants to look for. On the other hand, sample sizes are often too small, especially in rare diseases. So what we do is that we look in rare diseases for causal biomarkers, and then we just study the causal biomarkers in the entire population. So in other words, we make white data more to big data. So specifically, you have to see it in this way, that based on our feature selection algorithm, we select genetic variants as a proxy for a treatment, and then we look for a correlation to a biomarker, to a trait, you call it also an exposure, and then you look to a correlation again to the disease. And when there's a special relationship between these 2 correlations, then we can call it via causal inference, via Mendelian randomization, via propensity score, this as causal. And when you have found the causal biomarker, then you can check for genetic variants within this and look whether there is a good drug target, is the drug target causal for disease or not. biotx.ai is the missing piece in the tech bio ecosystem. So first of all, I call these data generating companies such as 23andMe. They're generating data. But as I said, converting high-dimensional data into knowledge, into products, is extremely difficult. It's not the same as in e-commerce. And another problem of these companies is that their data set can be quickly outdated by the release of the same data by public biobanks. On the other hand, I call them text mining by companies such as Benevolent AI, for example, in the UK. And actually, they screen scientific literature at scale, but in the end, they haven't found anything new in the data, and they're very vulnerable to data bias. Then we have protein modeling companies, such as Google's DeepMind's AlphaFold. And this is a huge success in the human science, because now we can grow in the chemical space. But there are no connection to the disease. And as I told you, the big problem in drug development is you have to pick the right drug target. And this is actually what we do at biotx.ai. We develop causal models of drug targets so that we can expect a high likelihood of clinical success. And that's why we do synergize with these new companies like DeepMind's, AlphaFold, because they broaden the chemical space and we choose the right drug targets. That's me and my co-founder and CEO, Jörn. He's a computer linguist. That's Charles. He's our CTO. He's a computational biologist. It's Christian. He's a former vice president at Medidata Solutions. And our strategic advisors, these are the Proteus Digital Health founders, Andrew Thompson and George Savage. That's Giancarlo Minella. He's a physician with a strong expertise in rare diseases. And of course, it's Jack Scanner. It's a former UBS investment banker. And he coined the term Irum's law about the decreased R&D productivity in pharma. And the only thing here, and this is really what we want to provide, we provide a better decision-making tool for the development of drugs, so better drugs can get faster to the patient. And with this, I would like to thank you for your attention.
Thank you. I have a question later on, but okay. First, the 4 minutes starts now for our Q&A session, and we'll start with Lamine here, and we will go on with Frank. Thank you.
What's your business model? Do you just focus on selling the tools, or do you want to develop own drugs?
This is what I answered in the first slide. So we have now a repurposed COVID treatment in a phase 2 clinical trial, and we have now one molecule with a new patent and formulation for Behçet disease. This is actually our product. This is our product development, but on the other hand, we also do service for smaller biotech companies, smaller pharma companies to provide this as a decision-making tool.
Okay, next question was by Frank, please. Yes.
I have not fully understood what the source of your data is. Is this already data from published clinical trial? Yeah. Do you need to access clinical trials? Because in published clinical trials, you do not have all the biomarkers you need?
No, the point already is we have now fantastic biomarkers. The problem not in Germany. For example, the UK biobank, you have 500,000 people completely annotated, so 11,000 diseases, and you have the full genome. And then this is just only the UK biobank. So we have now a database of around about 2 million genomes fully annotated, we curated over the last 5 years. We bridge the gap to the clinical stage by we use the genes, the genetic variants, as a proxy for the treatment. So the whole synthetic clinical trial is done within these biobank data.
Okay, we have a question here at the back, and then here again. Thank you.
So a lot of companies, especially in the US, are turning to like graph neural networks, more types of molecular modeling. I'm curious, as you evolve your platform and your feature set, how do you foresee integrating with those or the insights of some companies' system in order to add value to their clinical research process?
Yes, that's a good question. So, because I don't have a time here to explain in full detail our feature selection algorithm, but this is actually a knowledge graph. And the knowledge graphs gathered all these kind of information which is available from public sources or actually also from customers, for example, put it together, and then we create a kind of a shortlist which is then subsequently tested. And when there's a positive feedback, then there's an elaboration in this kind of space, which is somehow related in scientific literature, and it's then just kicked off and there is actually no signal. But the core technology is a knowledge graph for the feature selection algorithm. Yeah.
Great, we have another question here.
In bridging towards the clinical application, does your tool therefore avoid the in vivo studies, for example, or not yet?
Actually, good point. So what it brings in the end is that you have a better clue or that you have a better idea about the disease, and you know now the causal biomarker. And this is very important because when you conduct a clinical trial, you can put this causal biomarker also as an endpoint in your clinical trial. And this makes a clinical trial much more efficient, in my opinion, because it can lower the number of patients you need for your clinical trial. For example, when your endpoint is cholesterol level, it's much more easier to measure, rather than your endpoint is, for example, heart attack. Because heart attack, you need much more people, and the clinical trial has to pay much more longer that you have an idea. What do you mean?
Independent of the in vivo is still necessary.
But it's more efficient, because you have an idea of what you have to look for.
we want you next year. Okay, next question and last question, 20 seconds. Yes, no question.
Thank you. To clarify, like, what's your input? Is it the choosing of the biomarkers during the trial, or is it actually the choosing of the patients? Like, what do you do? What's your concrete input?
Yeah, I don't like it because we always see patient stratification and so on. So we have our database of 2 million genomes, and we now look what is the really causal trait.
No, was it the question? Was it with the patient or was it the?
What's your concrete input? Like, do you choose patients or do you choose?
You need both. You need both. You need
You need
people,
both. That's
and
great.
you need fully annotated patients. And the way patients.
Thank you very much.
Okay,
I'm sorry,
yeah.
I have to hold the yours.
Later, okay?
You need both. That was the answer.